In the previous video, we launched workflow, and now it's done. You can see that invocation is all green, and workflow produces two outputs, really, that we will care about. It produces a combined variant report. Looks like this. Sample, coordinates of the variants in the virus, depth, how many reads support this variance, allele frequency, and where it's located within the gene. It also produced consensus for all samples. Essentially, for every sample, it generates a sequence and changes nucleotide in this sequence if they correspond to variants, if these variants are above 50% frequency. So the problem with this is that, of course, these variants by themselves, they have no meaning. We need to analyze them. Oh, it's just a list. So in order to analyze them, we need to do two more things. We will need some additional data to interpret these variants. First, we will need a gene bank accession, a gene bank file for measles virus. The reason we will need that is because in this study, we focus on PVC locus. It's a locus which encodes three proteins, P, V, and C. And these proteins are encoded via overlapping reading frames. C is encoded in +1 shift, V is encoded in +2 shift after this RNA editing site, which occurs at position 2499. Now, the problem with this is that the tool that predicts effects of variants on coding region, for example, whether it's synonymous variant and non-synonymous variant, has a problem with interpreting coding regions that are this complex. So we're going to have to redo it ourselves. And this is also one of the ways to show how Galaxy can be used for conducting completely custom analysis. Let's begin with the gene bank file. So really, we need the gene bank file for the measles virus. And so that's the accession. It's right here in the variant list. We're going to go to Tools. I'm going to go to Get Data. And I will click on CBI accession download. Change that to direct entry because I want to input my accession number right away. And just ask it to give me the gene bank file. Just gonna click run. So this tool is actually designed to upload multiple gene bank files from NCBI. So what it generates not just a single data set, but rather it generates a collection. So it's a container. So what happens here is that this is the result, but the result is actually, it's like a folder with one file. But for our downstream analysis, we need to get that file out of the folder. So it's just a data set in our history. And the way we are going to do this is by going to Tools, selecting Collection Operations, and clicking on Extract Dataset. And really what I want to do, I want to extract the first data set from this collection right here. Click Run. Here it is. So that's the gene bank. This is the gene bank file we need. Done. So that's one. Okay, the next thing is that let's look again at our variants. So in this table, the only information about samples from which these variants are derived is this accession number. But we need more information. We need to know which country it's coming from, when it was collected, all that information. So we need not the sequences, but we need metadata about these samples. So let's get this data. One way of doing this is that when Galaxy was pooling data sets from EBI, it created a collection. And that collection, again, it's a container which has all this sequencing data. And each element in this collection is named after the sample accession. So let's just extract these things. For this, I'm going to go to Tools, to Collection Operations. and I will use tool called extract element identifiers of a list collection. And the collection I'm gonna care about here is this one. Just gonna click run. Now that we've got these IDs, we need to extract actual metadata about them. So for this, there's a variety of tools in Galaxy, and one I'm going to use is this one, FastQDL, which pulls this data from ENA. So here I'm just going to say this is a list of ENA accessions because that's the file that I have here. And I only need metadata. I don't need sequences themselves because sequences we already have. I already, in fact, use them in the workflow and so on. And I'm just going to click run. Now that metadata is extracted, I just have lots of files. Each file contains information about one accession. It's great, but I need to combine it into one data set so I can use it downstream. So I will go to Tools, select Collection Operations, use Collapse Collection to drag this thing into here. And importantly, I will select Keep One header line. Okay, let's look at the result. So these are the headers for the columns, and this is the actual data. So we are good to go. We can continue our analysis. To make it easy in our Jupyter Light part, let's label the data sets we need. So let's add tag. Let's call this run metadata. Let's call this gene bank. And let's call this variance. And actually, let's call this as well sequences. Okay. So we're doing downstream analysis. We will need our variance, the GenBank file, sequences of individual samples, and the run method.